Osmosis is the process of movement of water from higher concentration to lower concentration. And this movement takes place through a semi-permeable membrane or a plasma membrane. For this experiment, we will use a real large cell. Instead of using a model of the cell, we will use a real large cell. I am taking 2 plastic cups. In one cup, I'm adding tap water. In the other cup, I'm adding maple syrup. We are going to use a real large cell for this experiment, and that is the decalcified chicken egg. It's a single cell, and we want to add one of those cells in each one of those cups. But before we add this cell in the cups, let's take the initial weight. We are going to use a kitchen scale in order to take the initial weight and record it in the graph. After we have the initial weight, then we can add each one of those cells in the water cup and the maple syrup cup and start recording the time. We are going to measure the weight of these eggs on 15-minute intervals. After 15 minutes, I'm going to take each one of these eggs out from the cup. I'm going to dry them out, blot it on the paper towel, and then weigh them, and then record the data in the data table or on the graph that we have, and then put it back inside the cups and wait for next 15 minutes and so on. The egg cell that was in water had more water outside the cell than inside the cell. And the water goes from higher concentration to lower concentration. In this case, the water kept on entering the cell and the cell kept on growing bigger. And that's why it kept on increasing in weight. In the other example, where we had the cell inside the maple syrup, there was more water inside the cell and there was less water outside the cell, and the water goes from higher to lower concentration. That water kept on going from the cell to the syrup, and this cell was shrinking in weight, kept on losing the weight because the water was leaving the cell.